I am here with Professor Gordan Lauc, who is the foremost leader in the field of glycan biology, and he'll tell us more about what are glycans and what is this world of glycan biology. He's also the founder and the leader at a company called Genos, which is also a leader in this field in the world. And Genos actually just turned 18 years old. So Gordan, congratulations on this wonderful milestone.
Thank you. Thank you. No, it's normally companies would not celebrate 18th birthday. It's a kind of not a round number, usually to be 20. But I treat there's something like close to 60 people working in the company, and most of them are researchers. And for most of them, I was either their mentor for diploma work or a PhD, or my former students were their mentors. So I treat all of them as my kids, even grandchildren. And this is how I wanted to say that, they're becoming independent. They're now of legal age. They're not supposed to be minors. So now they're kind of ready to go on their own in the world. That was kind of my message, why I wanted to celebrate the birthday.
It's quite a symbolic birthday. It's quite amazing that a company grows so much and continues to lead the whole field for 18 years. But I'm assuming it did not start fabulously. It was probably very, very hard in the beginning, and every company's story is paved with difficulties and obstacles to be overcome. Could you tell me, if you bring me back to that time of the beginnings, how did it all start?
I would never say it was really difficult, because I'm still a professor at university. Actually, majority of my time I work at university. And I started a company 18 years ago as a kind of side project, which in meantime became the focus of everything what I do. And majority of things we do are still research, basic research, meaning we are funded with a research grant. So I think more than 50% of income are still the research grants. And we were growing mainly through getting more and better research grants and then translating this knowledge into the products and then putting products in a global market. So the challenging things was that I guess something like 90% of people or even more listening to this podcast do not know much about glycans. So we were kind of developing the field and trying to explain to other people that this is important. So these were the challenges. The company itself was, for me, always a pleasure, because I like doing research. And of course, there were difficult times. We didn't have money. We never have money, because whenever we get money, I spend it on hiring smart people and buying equipment. So we are always short of money. That's default. But it's a group of brilliant young kids who like to do science, and I really enjoy it.
That's quite amazing. You mentioned also that you were developing the whole field, and quite a few times in your presentations, I've heard you say that the glycans are quite important, but quite neglected because they're so difficult to work with. This machinery you need, the equipment, and I guess the expertise, maybe the protocol are very extensive and complicated. How did you have the courage to dive into something completely new? Did it start with some basic research discovery, or?
It's not completely new, of course. So the field of glycobiology is older than me, and I'm in it for over 30 years. And when I joined the field, there were people in it for 30 years already. So I'm not absolutely the pioneer of a glycoscience. But what we did first is we were the first to do high throughput glycomics, meaning studying a large number of people. So, for example, the first large study we did was in 2008, when we analyzed the first 1,000 people. And this was PhD of one of my former students. And only 2 years earlier, another student I had finished her PhD on something like 3 patients and 3 controls. So we made a quantum leap in the number of people we can analyze. And this was the largest analytical challenge. So something which works to analyze 1 or 2 people, convert into the methods where you can analyze 1,000 people's, have adequate analytical accuracy, all the statistics you need. This was a challenge. This was not easy to do. But we were kind of lucky that only maybe 2 years before we started doing genomics went the same way. So people started to do genome-wide association scans, analyzing a large number of people. And then we learned from the field of genomics, and when they learned how to use statistics, how to approach some of the technical problems, we applied it into genomics. So this is where we are pioneers, and this is why we developed this segment of a field. So not entire glycoscience, I cannot say that. But for high throughput glycomics, analyzing large cohorts, here we generate approximately 80% of global output. So my lab does 80%, the rest of the world does 20. So it is a quite dominant position.
That's quite amazing. And what I really meant was this pioneering in the field of high throughput glycomics. That's still a big, as you said, it's a huge leap. And from that first PhD student who had like 3 patients, 3 controls to like 1,000, what happened in between? Was it some kind of big discovery, or was it just a big trend that was warming up in the sciences?
It's a combination of things always. So it's analytical progress. So the machines were getting better. They were not developed for glycans, but we modified them to be used for glycans. And the great success of the large genomic studies, where we really started to understand the function of individual genes by looking through differences in different people, made me realize that there is a huge potential. So many people still do not understand that. They don't know why I want to analyze millions of people. They think we should focus on the details of a structure in a small number of people. So, of course, not everybody will agree. But I think the 2 things: analytical progress, which is happening all the time, and the realization that by doing this type of research, we could really learn something new. And we did learn a lot. For example, majority of people will still not agree with me, but the key differences between individuals are in protein glycosylation. This is what is making us different. And also, glycosylation is what enabled life to become multicellular. Without glycans, we would still be a single cell organisms. So, and there is a great structural complexity. So even after analyzing, I think it's over 300,000 people that we analyzed so far, we learned a lot, but we learned much more about how little we know. And I think it's a general principle. The more you know, the boundary to the unknown is larger, so you know how much you do not know. So I think our knowledge about biology and medicine in general is still very rudimental.
I want to dive into the complexities of glycans and all of the implications they have. But let's start with the beginning for anyone who's not familiar with glycans. Maybe best to hear it from you. I don't want to burden you with the burden of defining glycans, but I'm going to have to do it. It's like making Mick Jagger sing one of his hits. So what are glycans?
It's relatively simple. So proteins are molecular robots which do majority of work in organisms. And majority of proteins are glycoproteins, meaning they have a glycan part and a protein part. And glycans are not directly encoded in a gene. So the structure of our protein is defined by our gene. So we have a sequence of nucleotides, which we inherit from our parents. They define the structure and function of a protein. To the contrary, the glycan part is encoded in network of hundreds of genes, epigenetic regulation, environmental factors. So it's a highly dynamic network information which generates the other aspect, the other part of protein structure. And more or less all processes which developed after we became multicellular involve some aspect of glycosylation. I can tell you one example. For example, immunoglobulin G. That's our major weapon in our immune system. It's used antibodies. They're used to recognize the pathogens and eliminate them. Also the tumors and so on. And their effector functions, meaning what are they going to do when they see an antigen, is determined by a glycan. So if you have one type of glycosylation, antibody is pro-inflammatory, it's promoting inflammation. If we change glycosylation to be a little bit different, it's suppressing inflammation. So exactly the same molecule, the same protein part, the same gene has totally opposite functions depending on what glycans we add there. And the decision which glycan will be added is a very complex one. So we map the network of over 100 genes which work together to decide what kind of glycan to put and when. And this is something we can change. Even when something radical is happening in an organism, in a few days we can completely reglycosylate all our IgG proteins and make them work in the opposite direction. It's more or less it's a secret of life, multicellular life, which majority of researchers are still ignoring. So we are kind of picking the low-hanging fruit and finding the most interesting things at the moment.
So in some of your papers, I'm seeing a lot of research focusing around how patterns of glycosylation are changing in different cohorts and in different countries across age. Can you tell me more about how do they change with?
So what we have noticed very early when we started to do large cohorts is that if, for example, we compare patients and controls and do not take an adequate care to have exactly age-matched controls, we always have the change going in the same direction. So age is practically the strongest factor affecting the change in immunoglobulin G glycosylation. And it's making it probably too technical, but it has less sialic acid, less galactose. And this is a change which we see with age, but we also see it in the majority of diseases. And this is a change which correlates with the low-grade systemic inflammation. So we know that inflammation is the driver of majority of complex diseases of today. It strongly associates with obesity. It's associated with cardiometabolic diseases, including diabetes, cardiovascular disease, and so on. And glycans change in the same direction as we age, but with a different pace. In some people, it will go slowly, and in some people, it will go faster. And people where we have a faster change have much higher risk of developing disease and eventually even dying than people who have this slower type of change. This is something we named the Glycan age. So you can have your chronological age, meaning this is the number of circles around the sun which our planet made, which is totally arbitrary number because it's, age is not. If you move to Mars, 1 year would be something completely different. And then we have a biological age, which is the molecular changes which happen through time, which have an individual pace, and the pace which we can affect with the way we live our life, even with the way we think.
There's a lot of these biological clocks nowadays. It's quite a hot topic at the moment, and we talk about it in the lab a lot, even though it's not our primary field. What difference I noted between the other systems in which biological clocks are implied and have been developed by other people and glycans is that it's actually one difference that you mentioned in one of the podcasts, that glycans are actually also effector molecules. So if you reverse the biological age from, let's say, from another system or another biological clock, doesn't necessarily mean you really become younger or that your system has changed. But in glycans, that might actually be true, because if you actually change your glycosylation pattern, you might actually do something to the whole organism health-wise. Could you speak more about that? Because I'm still unclear on what is the effector role of glycans and how would, let's say, reversing your biological age from 70 to 50, how would that really affect the organism besides pro-inflammatory and anti-inflammatory?
Maybe a few words about the clocks before we go more into functional aspects. So there are more or less two classes of molecules which are basis of different clocks which are being developed, aging clocks, either the DNA methylation or protein glycosylation. Recently, there was some kind of a proteomic clocks, just a concentration of proteins, or some kind of blood-based biomarkers. But more or less, glycans and epigenetics are the ones which large studies have shown to be mostly correlated with age and disease. Epigenetic clocks have been developed in a way that you take some aspect of biology you want to look at, either the chronological age or diseases or whatever you know for a large number of people, and then quantify now over 600,000 methylation sites using the EPIC arrays, and then you model which of these 600,000 are the best predictors of the variable you're interested in. So they have no ways of knowing which of these CPG sites are functional. And for example, I spoke to Steve Horvath 2 years ago. We organized the 10th anniversary of the epigenetic and glycan clocks. And he said, yes, I did select this 300 and I believe 73, but the addition, another 373 would be equally good. So individual ones are not really functional. They're just information. Some of them may be functional, but we don't know which of them are. For glycans, we know what I mentioned a little bit earlier is that different glycan structure on the immunoglobulin G will direct IgG to different receptors. And the most known example is something called the core fucose. Core fucose is blocking the activating receptor in the natural killer cells, and it's preventing immunoglobulin G from initiating antibody dependent cellular cytotoxicity. So one of the functions of antibody is, for example, if you have a cell which is became a cancer cell or it's infected by a virus, antibody binds to the cell, NK cells come and kill the target cell. It's called the natural killer cells, eliminating the infected or the tumor cells. And majority of our antibodies, over 95%, have this core fucose, which is blocking the interaction between antibody Fc gamma receptor 3A. And it has to be that way because otherwise the NK cells would randomly attack our cells and there will be a lot of autoimmune damage. So most of our antibodies are they have a safety switch against ADCC. And if you remove the core fucose, it becomes more than 100 times more potent. And now there's a new generation of monoclonal drugs. So, most of these smart wonder drugs are antibodies. And until very recently, nobody was thinking about glycosylation. So even the antibodies which were supposed to activate ADCC, they had a core fucose. So you had to go with 100 times higher dose than needed. Now they developed the glycoengineered antibodies without a core fucose, and they're much more effective. So they can work in a much lower dose. So that's one of the aspects, how glycosylation affects antibody function. There are multiple others. It was shown that cellulation is suppressing inflammation. And for example, if you take immunoglobulins from young people and inject them in the older people, they all feel much better because you have a suppression of the low-grade chronic inflammation, which is wasting huge amount of energy in the old people. So I know a couple of people who are doing regular IVIG every autumn just to get through the winter. And you think about Brian Johnson, who is a kind of a prominent figure and is experimenting everything. He's replacing his plasma with the albumin and IVIG. So he's suppressing inflammation using globulins without even knowing that it's glycan parts, which are one of the key elements for suppressing inflammation.
When we talk about suppressing inflammation and all of these effector aspects of glycans, are we only talking about IgG or are we talking about many other molecules as well and just using IgG as a proxy?
So IgG is different than most other molecules. I would say IgA, which is another immunoglobulin, might have a similar role. Other proteins have different functions for glycosylation. So this suppression of inflammation is immunoglobulin specific thing.
There is this paper, and I want to credit the first author. His name is Jerko Stambuk. I hope I got that right. And the paper is called Global Variability of IgG Glycome. And there's this beautiful figure 1, which is showing, I think, cohorts from 5 or 6 different countries all over the world and showing glycosylation patterns with age in males and females. And it's showing that beautifully it goes up. There's 3 points that are demarked: 30 years old, 50 years old, 70 years old. And in men, it goes quite, like a straight linear line, repeatable, well, not repeatable, but very equal steps at every point. Whereas in women, quite a few of these, and it seemed to be quite consistent among different country cohorts, there's this either dip or jump at the age of 45, 50, which is the age of menopause. What do we know about glycans in women, and in particular in this age, and how does it affect their health, and what can be done about it, and so on?
So the study which Jerko did was actually comparing 27 different cohorts all around the world. So we were kind of looking at the country-specific effects. We did a number of other large studies of the with the more samples in this period of perimenopause, and the effects are extremely strong and very clear. So before the age of menopause, women generally look younger than men, meaning they have glycans which are more suppressing inflammation, and it's a consistent with general knowledge of biology. So in the reproductive age, women are more resilient to kind of everything because that's evolution. They have to survive and bring the kids. But as one of the first symptoms of menopause, which happens even a number of years before the official menopause, glycans have a rapid acceleration in age. So we've seen examples of women who would age kind of 30 years within a 6 months or something like that. And this is mainly driven by estrogen. So we did the number of studies, even properly placebo-controlled studies, which show that the estrogen is one of the key regulators of IgG glycosylation. Interestingly, in both men and women, in men, it's also estrogen. So when people try to kind of improve health of men by adding them testosterone, and then if they don't get the levels of testosterone they want, sometimes they add aromatase inhibitors, and aromatase is enzyme which is converting testosterone to estrogen. They increase testosterone, but they block the beneficial effects of testosterone. Part of the effects goes through estrogen, and this anti-inflammatory effect goes through estrogen. So this is one of the priority fields of research for Glycan, which is our spin-out company to commercialize these markers. And this is the early prediction of perimenopause and also the personalization of hormone replacement therapy. Because this huge increase which happens in perimenopause can be reversed by the hormone replacement therapy. But each person is different and things have to be personalized. And there are a couple of large clinics globally which now actually use Glycan to optimize hormone replacement therapy, to personalize it, to make it kind of adequate for a given patient.
So did I understand correctly that the dip or the big change in glycans happens before the menopause?
It happens a couple of years before. So the menopause is defined as 12 months after the last bleeding. So once you have your last cycle, you have to wait for a year, and then you are declared menopausal. If you have another cycle, anytime within that year, you have to wait for another year. So menopause has a very kind of horrible definition. It's not defined at any molecular marker. It's defined by 1 year since your last cycle. And symptoms often start 5 to 10 years earlier. And we see changes in glycans even then. And even we see them often. Of course, not everyone is the same, but often we see them before any other symptoms of perimenopause.
Would there be a potential in delaying menopause with glycans? Maybe a bit of a too far-fetched of an ambition?
So with I wouldn't go that far because I think that the reproductive cycle of a woman has other regulatory mechanisms. So glycans here can more or less be used as a biomarker so that you see what is going on. And we claim, actually filed a patent for, that IgG glycom composition can be used to estimate the concentration of estrogen in the last couple of weeks. So it can be kind of analog to HbA1c, glycated hemoglobin, which is used to estimate the level of glucose overload in the last few weeks. For estrogen, you could use IgG glycom. Because one of the problems in perimenopause in women is that the hormones fluctuate rapidly within a day, between days. So if you just measure at one time point, you have no relevant information. So for example, one of the kind of quite a lot used test today, so-called Dutch test, where they collect samples for 28 days and then average. If you want to measure something and then you have to take sample for 28 days, that's a nightmare. But women do it. And IgG glycom is doing it for us because IgG glycom is under control of estrogen. It's continuously being produced. Take a single blood sample, and you can kind of estimate the average estrogen concentration in the last couple of weeks.
What kind of potential do you see with determining the glycan age and giving all these proxies when it comes to these bad effects of menopause that women experience? Is there any
So I
therapeutic?
think The
potential
first
benefits?
first benefit is early information. So usually in menopause is kind of taboo. Everybody is afraid of mentioning it. Everybody is afraid of this happening because often women think they're not women anymore when they kind of lost of a cycle and reproductive power. So there's very little research. It's heavily under research field, and it's in many cultures it's still just not talked about. So women often do not recognize the problem. And I read some statistics. I think in UK they spent 7 years and I don't know, maybe 100,000 pounds until they got to the right diagnosis. Because usually when they go with a symptom, they get diagnosed with a depression, fibromyalgia, whatever. And they're completely wrongly treated because they treat the symptom and not the problem. So if you realize that it's a hormone deficiency, which you have as a problem, then you can act and supplement the hormones in a proper way. So one thing is early diagnosis. The second thing is we don't really know what are the normal hormone levels for a given individual. We have relatively wide normal ranges, but being within the normal range doesn't mean everything is okay. So I'm not claiming this is the best and the only way we should be used, but this is something that can be done today is kind of titrating the hormones to see a beneficial effect on a glycan age. Because we know glycan age is estimating estrogen concentration, and you kind of add estrogen and you use this as a kind of tool to personalize it, because you can give exactly the same amount of estrogen to two different women and get completely different levels the second day. So it's biology is different. So that's one aspect. And the other aspect is it's not only estrogen. For example, there are some clinics which also add a little bit of testosterone to women, because testosterone is not only male hormone. Women also need testosterone to build muscle and so on. So it's another layer of information which can be generated, which well-educated physician can use to personalize therapy. Of course, the problem here is, the moment you start interfering with somebody's health, you could also be doing damage. So things has to be regulated. And then again, there is this huge anti-aging industry, which is completely unregulated, especially if you talk about the US and anything even topical. So I don't know whether you know, but in US, you can put anything in a cream they're putting on body. There is no regulation. What are you putting? You could put botulinum toxin and kill somebody and it will be legal because you put it on the skin. In Europe, it's more complicated. So now there are some companies which are developing very aggressive chemical treatments which are given topically. For example, you cannot give rapamycin to people legally, but you can make a cream with rapamycin and apply it to the large surface of a skin and then have a systemic effect of rapamycin without any regulatory approvals. This is one of the areas where we are kind of positioning glycan aging is kind of at least let's monitor the effect. Let's see what happens when you do something.
I've heard you say that your glycan age is actually quite terrible, and your experience was that you tried fixing it, you tried hiking, and hiking didn't really work, and other things didn't work, and that the only thing that worked for you was diet. But for other people, it might be some of these other core physiological things. So could you speak more about this individuality of how glycan age changes in it?
Yeah, more or less, there are 3 or 4 key aspects. One is diet, one is physical activity, and the third one is something we can say either stress, psychological well-being, or whatever you want to call it, including sleep and everything that is kind of coming from a brain. And the only thing, well, the problem is, everything we know in medicine is based on taking a couple of hundred people, measuring something, taking an average, and then making a decision whether something works or not. And it's very hard to identify molecules or procedures which work in a 5% of people or 2% of people or something like that. And if you look back in the studies, everything what was done so far, not everything, but nearly everything, something which regularly works is a caloric restriction. So eating less makes us healthier in all aspects. But I think the reason why we are getting this type of a result is that majority of us are eating too much. So it's not that eating less is good, but eating too much is bad. And by not eating too much, we see a beneficial effect of not eating. So this is why diet helps a large number of people. Regarding physical activity, what we learned after I said what I said then is that it really takes a long time for physical activity to show beneficial effects. We had 1,000 people put on relatively aggressive exercise program, middle-aged couch potato type of people. And after 3 months, majority of them got worse. After 6 months, majority of them were still worse. Only after 9 months, we started to see more and more people get better.
And when you
And
say,
then
sorry, when you say worse and better, you mean in terms of glycans or
glycan
in terms of, okay.
in glycan age, because glycan age is one of the universal ways to quantify low-grade systemic inflammation. So they were more pro-inflammatory. And and we know that, if you exercise too much, everything hurts. Your feet hurts, your muscles hurts, it hurts. So there's something going on which is not good, because pain is a signal that something is not good. And just recently, I think last year or maybe year before last, we published a paper where we compared sedentary people, regularly active people for years, and professional athletes. And people who were regularly moderately active, they were much better. Professional athletes were actually worse than a sedentary people. So too much physical activity is not good. We need prolonged periods of activity, which doesn't have to be very high intensity, and then we see beneficial effects. So probably what I was doing wrong, and this was experiment done more than 10 years ago, I was hiking too much. So probably doing a little bit less intensive, more prolonged activity would be better for me. But for me, actually, the main problem is in my head. I'm always running too quickly, too many deadlines, working too many things in parallel. So I'm just living too quickly, and I can't change that, because then it's not me. So I kind of accepted the risk, and we're just about to publish a new paper about mortality risk. And I think the figure is, for men, every glycan age, increasing glycan age, is something like 10% higher risk of dying in the next 10 years. But of course, 10% on the basic risk, and the basic risk is something like 3%. So you're 10 years older in glycan age, instead of 3%, you have 6% of dying, or something like that. So it's not horrible, but it is there.
Yeah. When you mentioned that you compared, I find this study very interesting, that you compared sedentary people with moderately active with athletes, and that athletes were showing quote unquote worse result. Can we actually say, because if we take this glycan age as a proxy for inflammation, like let's say it's a low grade inflammation proxy, can we actually say that they are worse, really, like physically? Because there's always this question of maybe a little bit of inflammation is good, maybe a little bit of pain is good in certain contexts and so on. So it's probably also a different kind of inflammation in exercise compared to like being very sick from a virus and so on. So how do you
So
distinguish these?
I'm okay. When I say good and worse or better, I'm just talking in the aspect of glycan age. So especially when people are young, a little bit of inflammation, building muscles, it's definitely not a health risk. So I'm not saying that more physical activity is necessarily bad, but it is increasing the low-grade systemic inflammation, and it always has some negative aspects. And we did work with a lot of professional trainers, and some of them managed to kind of have all the beneficial effects of training, but avoid this inflammatory side effect. And these guys were usually combining sufficient relaxation periods, maybe cold baths or something like that. So something to balance the high intensity training, because definitely too much exercise is better than too little, but maybe we can also optimize it. Maybe we can see what works really for me, and there is no professional trainer which can say just by looking at you. They have to quantify something. They have to measure what is going on molecularly, because people are different. So I would say, I don't like to kind of claim that everybody should be out doing Glycan age, because at least I'm selling the test, so I'm making money out of it. So it's it's not up to me to say that somebody should do the Glycan age, but I hope relatively soon we'll have it as a part of routine healthcare. And then by monitoring IgG glycosylation, one can get insight into this low grade systemic inflammation going on, and then try to control it, because it is doing damage. There's no aspect that inflammation is doing damage.
Speaking of the test at Glycan age, how does it work? That my understanding is that you get a little kit, you might get a little as little invasive needle as possible. You leave a little drop of blood and you send it back to the company. Company analyzes, here's your Glycan age. Is that how, first of all, does that how it works? And then what happens next? Is there a personalized plan or recommendations on how to reverse it? What happens
There,
after that?
there are two ways of doing glycanage. One is the way you just said. You go to the website, you buy a sample collection kit, you do a finger prick, collect your drops of blood, send it back, and then you can get, actually, I think have something like half an hour of consultations included in the test, where you talk to the professionals in Glycan age who worked with thousands of people so far, so they can tell you what could you do. But personally, I prefer when this test is done within a medical setting. And now there are over 2,000 clinics all around the world in literally 60 countries, something like that, where you can also do Glycan age as part of other tests they do, mainly for this longevity, disease prevention, and so on. Because when you have a half an hour consultation with a specialist, that specialist doesn't know everything about you. And if you have a good physician and physician knows everything about you, all other blood tests, all other functional tests, then it's much easier to interpret this information in a right sense. Of course, the problem is that the majority of physicians do not know anything about the Glycan age. So it's very hard to use it, but I think this is changing. I hope this year at Longevity Fest in Las Vegas, we'll have entire session about the glycans and education about glycans. We are currently talking about it. So that more and more medical professionals who work with patients will know the meaning of these values. So for now, it's either one of these 2,000 clinics, or if you're an enthusiast, you want to do it yourself. Yes, you buy a kit, you do it, you talk to a specialist, you read, and then you try to do experiment on your own.
My field is the field of neurodegeneration, and this is what I'm doing my PhD in. And I read recently about a case of a woman who had, she's from a Colombian family, and her family history indicated that she should really get Alzheimer's disease because she had multiple mutations that should be predisposing her to this. However, she lived well into her, I believe, 70s, maybe even 80s, and she never developed dementia or anything like that. Have you ever had any anomalies with glycans like that, where a person has maybe a terrible glycan age, but actually nothing ever bad happened to them, or they look amazingly healthy, and or in the opposite direction?
Well, yes, of course, because there are many aspects of life which affect each of us. And for example, one of the worst drivers of glycan age is obesity. And recently, some time ago, we had an example of a person who was pretty obese, something like 120, 30 kilos. And he wanted to do a Glycan age, but he was more concerned about his friend who was something like 180 kilos. So two of them did the test. And actually, this really obese guy was better than the one who was much less obese. Although one would say this morbid obesity is definitely horrible and it's good, not good for your health. But the Glycan age was not that bad. And we know that something like 40% of your Glycan age is genetic, meaning you just have good or bad genes for that aspect of biology. The rest is different life factors, and obesity is one. Maybe somebody can be obese, but he's happy, he's good, he's a little bit physically active. So maybe he will live into his 70s and be relatively okay. So, of course, we always have extremes, and with the more people being sequenced, we know that some of the well-known disease-causing mutations will not cause diseases in some people. They compensate in some way. So, we are on this planet because in the last couple of billion of years, our ancestors survived everything. So we are optimized for all kind of problems, all type of complications, and we survive. As I said at the beginning, we know so little about biology. And I think we have to be modest and admit that we really do not understand biology. And we will always see this type of examples. People with mutation healthy, people in the horrible glycan age healthy, people with the great glycan age developing disease and dying. It's never 1-0. It's always just a risk. But we have to accept that, statistics is real. If there is a 15% higher chance, there is a 15% higher chance. It doesn't mean it will happen. It's just higher chance. And it's better to have a lower chance of dying than higher.
What attention have you devoted to understanding the biology of how different organs, or maybe in particular diseases, change their glycans? So just maybe adjacent to the neurodegeneration, do we know anything about how the brain changes when it comes to glycans? And there's always this topic of low chronic inflammation in different degenerative diseases.
So we haven't done much in the neuroscience because, I'm afraid of more or less two fields. One is immunology, which is horribly complex. The other one is neuroscience, which is horribly complex. So we have one of my postdocs that's focusing on neuroscience. We did a couple of interesting studies with brain development, evolution, comparative glycosylation, different organisms. We haven't done much on neurodegeneration. Just a couple of days ago, I discussed with a colleague, we never did an Alzheimer cohort. That's stupid. We have to do a large Alzheimer cohort to see, because I'm pretty confident we'll see a signal, because whether it's energy metabolism or whether it's inflammation thing, we'll see a signal. So yeah, this is something which is ahead of us. We have to do that. Different tissues are of course different. We even did some kind of for example, when you differentiate the stem cell into the neuron, you see the specific changes in glycosylation. In organoids, we see some effects. So it's definitely it's an aspect. Well, to make it simple, all the membrane receptors are glycosylated, and their glycosylation affects their function. One of the first examples where the glycosylation was shown to be functional was the notch, which is one of the key regulators in brain development. So it's a huge unstudied field which has to be addressed eventually.
Speaking of fields that need to be addressed eventually, and I'm assuming Genos and your group will play a big role in it. What are the immediate next steps on big studies you're aiming to do, or big cohorts you're aiming to analyze?
Yeah. So at the moment we have 9 Horizon Europe projects, each of them having maybe 10 partners on average, a huge cohort. So we'll continue doing 50 to 100,000 samples per year. We try to get them as interesting as possible. But what we are currently doing, we are trying to spread our technology to as many labs as possible. So now we have a Glycan profiler, which is a dedicated machine. We have a kits, which will also be a CE-IVDR compliant. So people can actually go buy a machine, buy a kit, do it. We are developing the databases, the Human Glycan Project databases, where we'll try to put as much glycan data as possible in a public domain so that other people can start working for it. Because I remember very vividly in the is how molecular biology was unreliable and how things started to progress only when the kits became reliable and when the good databases were developed. And I think this is where I will personally focus the most in the next couple of years, developing good databases and developing good analytics that as many labs as possible can relatively easily start working on glycans.
In all of these years of developing all of these amazing technologies, doing all of these projects, and you mentioned a couple of times that the number of partners that you do and the cohorts that you actually select are quite amazingly well documented. What was the one or two most spectacular results that you still remember that you went, wow?
So many of them. So I think one of the most interesting thing is how we are trying to understand how glycosylation is regulated. And for a long time, people were thinking it's expression of glycosyl transferases, meaning enzymes which build them. It is not. It's much more complex network of proteins which regulates protein glycosylation. And you can have the same protein with a completely different regulation in a different parts of the molecule. No, FAB, FC part of IgG have different regulation of glycosylation. So I think that complexity is one great wow, which we are still looking at. And maybe the most interesting discoveries were that when you look in people 5 to 10 years before develop disease, we already see a change in glycans. So you actually take a person, analyze glycans and see, look, you're on your way to diabetes. You're on your way to heart attack. And then by doing lifestyle intervention, this risk goes down. So you can actually monitor individual's path to the disease. Or something really wow, which we haven't published yet, is that if you go fasting, meaning either 0 calorie or maybe up to 200 calories a day, your glycans on IgG would start to change in 3 to 4 days, which we don't know the molecular mechanism how this can happen, because IgG lives for 3 weeks. So how can it change in 3 days? But it happens. It happened in 3 cohorts so far. We replicated it perfectly. So there are mechanisms how we can really reglycosylate IgG very quickly in fasting. So it's, yeah, maybe these two, but there are probably more. I just forgot. I think I had close to 60 people in my group, 28 postdocs. And, every week I hear one or two new things happening and developing. So for me, it's a roller coaster, just something going on and happening and trying to understand, which is fun, which is great. Because, when somebody is at your level, it takes a long time to get some results. You have to work for weeks, for months, and then you get answer to a question you're trying to address. For me, it's coming twice a week. So it's a great
That's quite
position to
amazing.
be. Yeah,
Well, like
it's
watching
really.
YouTube at 2x. So I think there's obviously a huge potential in all these big databases you're creating and sampling so many people and so many different cohorts cross countries, cross continents. Are you also building some kind of a system to follow up with those people? Because there's obviously a potential to, in a certain number of those populations, some people tick off the box that they got a certain diagnosis. Then you can focus on that and maybe try to develop a prediction for that disease and essentially use this to predict any disease.
So this is painful, meaning waiting for something to happen. So we try to do another approach. And this is when I find an interesting paper in the literature, when somebody did it already, meaning somebody was collecting samples for 20 years and found that the protein X is predicting something. I send an email and say, would you like to add glycans to that story? And this is how we did the most interesting things. For example, I sent an email to Mike Snyder. Mike, can we add glycans to what you are doing? We did it. We added glycans to there's a celery trial, which is people who were 2 years on low calorie diet. There's a heritage done 30 years ago where people were it was not 30, it was something like 20, very carefully followed for physical activity. There are fantastic samples in the fridges all around the world, collected 30 years ago with a clinical follow-up, and then you can address specific questions. So that's a kind of easy route to what you are saying. We, of course, doing what you said too. So Glycan Age is accumulating data, sharing data with the clients. Some of the clients are interested to do research, but this will take, you have to wait for people to get sick to get that feedback. And if you have samples collected 30 years ago, it's easier.
I know very few people who are running a team of 25 and more, let alone 50 and more. How does that work in your daily life? How do you organize it, and how do you try to lead your team?
Yeah. So it's a little bit unique situation because I own the institute. So some of these people are with me for 15, even 17 years. So in any academic setting, they would be full independent. In Genos, they're also nearly full independent. So I don't manage 50 people. You cannot do it. There's no way you can do it. I barely know the names of some of the people. So I work with a team of maybe 15 seniors who then kind of have their own groups. So we do function as one team because we all focus on glycosylation, different aspects of glycosylation. We have joint meetings twice a week, entire group. But personally, I don't manage them. So and I'm not the micromanaging guy. I always tell them, come to me only if something is not working. If everything is fine, you make your own decision, you move forward, and come to me when you have something interesting.
That's amazing. But still, even 15 seniors, that's a lot of briefing to do. How do you organize your day with
I
all
do
those
that.
people?
That's the only thing I do because I literally I'm not writing any. Occasionally, I write a paper as a main author where I did do most of the writing, but I now mostly do editing and fixing. I don't do even entire write ranting. I don't do reporting. I just talk to my team and collaborators and people like you. I'm a kind of I don't know what am I. I'm manager slash PR agent slash spokesman.
I've heard you say that recently you've started gardening a little bit to decompress and enjoy your life. Is that
Yeah.
something you're still practicing?
I'm in a garden at least 2 hours a day. I woke up at 6 today and I was in the garden before we started recording this. So and the way I do it, it's kind of gym. So I don't do everything in the easiest way. I kind of do some movements and positions which are a little bit hard. So I said both it's a gym and it's a production of food, which is a huge problem today. Food is the worst poison. So having a source of food that is not treated with anything, it's important. So I want my kids to eat healthy, so I try to produce food.
That's amazing. All right. So I'm struggling still to find appropriate ending questions. And I want to end on two, actually. Obviously, I want to hear your advice for the young scientist out there struggling to build a niche of their own. So maybe you can tell me that. For someone in the beginning of their career, in the beginning of their master's or PhD, what's one piece of advice you would give them?
I think the key important is select your mentor carefully because the worst thing you can have is to work with somebody who is not supportive of young people. And I've heard so many colleagues saying something like, I burned 3 postdocs on this project, which is horrible. You hire the 3 bright persons, force them to work on something which is very difficult, and then you just kick them out. I hate that. So I always treat my students as my children, and they treat me like their father or grandfather. So I really have good relations with most of my former students, and I have many. So I think that the key thing is see with whom you want to work. So the easiest way is check what happened to their previous students. How did their careers go? How did they publish? That's the most important thing. The topic is less important because when you're a student, you don't really understand the field. And then you like the topics where you have an interesting professor, somebody giving good presentations. You think the field is interesting. But whatever you do, when you learn enough about it, it's becoming interesting. So I started my career with the physical biochemistry. So I was studying binding of proteins to some kind of small groups, and I moved into glycoscience. So I think instead of focusing on a topic, and especially young people, often they make that mistake. They say, "Oh, I did diploma on I don't know, tumor biology. I want to do tumor biology in the future." I think that's wrong. Find a good lab, good mentor, and that will help your career and definitely your quality of life. So that would be my advice.
In the beginning, and this is something I want to finish with. In the beginning, you mentioned when you just started talking about glycans of this conversation that many people disagree with you on certain aspects of your views, be it on biological clocks or glycans themselves. Do you feel like you are a contrarian in heart? And are there any topics in which you really go against the mainstream of science or what is currently trendy or hot?
So I'm not a contrarian. So I do kind of comply with agree with the majority of people on majority of things. So I'm not the one who just wants to be different. But there are some aspects of science which I dislike. And the first, I think our publishing system is completely broken. The way we report research, the way we promote research, it became horrible, and I dislike it strongly. And the second thing is many researchers are under heavy influence of their funders, especially the pharma industry and the food industry, and a lot of research is just fake. So it's a frustrating to see bright young kids coming with enthusiasm and then hitting a wall of maybe the COVID was the best example, where more or less the farm industry bribed some of the key institutions and shifted entire field into something which was simply not true. You do not enforce stupid restrictions on billions of people without any evidence that they work. You don't do it. We did it. Or you do not mandate experimental vaccine on billions of people. You don't do it. You do it for maybe high risk groups. And it's, we are paying huge price by the loss of trust in science. What is happening in US now is horrible. They're practically shutting down NIH because it's, of course, there were some bad things being done there, but now we have people ruling the largest economy in the world who are really anti-scientists. And it's science who brought it upon ourselves. So I was the contrarian in the COVID period. I was kind of always fighting this propaganda, which was not easy. Yeah, but that's fortunate that passed.
Okay, I'll make sure in the future not to end this with this question, because now I want to talk about this for another hour. But I feel like that could be a good reason for us to talk again. Professor Gordan, thank you so much for being the guest.